Receptor desensitization, cell signaling network, and impairment of signaling mechanisms. Receptor desensitization. Receptor desensitization refers to the process by which a receptor becomes less responsive to its ligand after prolonged or repeated exposure. This phenomenon is crucial for maintaining cellular homeostasis and preventing overstimulation of signaling pathways, which could lead to adverse effects such as inflammation or excessive cell growth. Desensitization can occur at different stages of receptor activation, including receptor binding, signal transduction, and cellular response. Mechanism of receptor desensitization. Receptor desensitization can occur through several mechanisms, including phosphorylation. One of the most common mechanisms is the phosphorylation of the receptor by kinases, such as G-protein-coupled receptor kinases (GRKs) or protein kinase A (PKA). Phosphorylation alters the receptor's conformation, reducing its ability to bind to its ligand or interact with downstream signaling proteins. Receptor internalization. Another mechanism is the internalization of the receptor from the plasma membrane into intracellular compartments, e.g., endosomes. This process effectively removes the receptor from the cell surface, decreasing its availability to bind ligands. Receptor regulation. In prolonged or chronic exposure to a ligand, receptor downregulation can occur, where receptors are degraded in lysosomes, leading to a decrease in the overall number of receptors available for activation. These processes prevent overactivation of signaling pathways, which could otherwise lead to harmful effects such as chronic inflammation or cellular damage. Examples: beta-adrenergic receptors. These receptors, which mediate responses to adrenaline and norepinephrine, undergo desensitization following prolonged activation. Phosphorylation by GRKs leads to receptor internalization, reducing the effectiveness of adrenergic signaling. Insulin receptors. Prolonged exposure to high levels of insulin can result in desensitization of insulin receptors, which is a key feature of insulin resistance in conditions such as type 2 diabetes. Cell signaling network. The cell signaling network refers to the complex system of molecular signals that coordinate cellular responses to various external and internal stimuli. These networks involve a series of interconnected signaling pathways that regulate processes such as cell growth, differentiation, metabolism, apoptosis, and immune responses. The signaling network is highly dynamic and involves a large number of receptors, signaling molecules, and intracellular proteins that work together to propagate signals from the cell surface to the nucleus. Types of signaling pathways. Receptor tyrosine kinase (RTK) pathways. These pathways are initiated when ligands such as growth factors bind to receptor tyrosine kinases on the cell surface, activating their kinase activity and leading to phosphorylation of intracellular substrates. This triggers downstream signaling cascades that regulate cell proliferation and survival. Example: EGF epidermal growth factor signaling pathway. G-protein coupled receptor (GPCR) pathways. GPCRs are involved in a wide range of physiological processes. When a ligand binds to the GPCR, it activates an associated G-protein, which then modulates the activity of intracellular signaling molecules such as CAM, IP3, or CA2+. Example: beta-adrenergic signaling. WNT beta-catenin pathway. This pathway is crucial for regulating development and cell fate decisions. The binding of W-ligand-frizzled receptors inhibits the degradation of beta-catenin, leading to its accumulation in the nucleus and activation of target genes that regulate cell proliferation and differentiation. Notch signaling. This pathway controls cell fate decisions during development. Notch receptors bind to ligands on adjacent cells, leading to the cleavage of the receptor and release of the intracellular domain, which translocates to the nucleus to regulate gene expression. Integration of signaling pathways. The cell signaling network is characterized by crosstalk between different pathways. This crosstalk enables the integration of multiple signals and allows cells to respond to complex environmental and physiological conditions. For instance, signaling through PI3K-AKT and MAPK pathways can be integrated to regulate both cell survival and cell cycle progression. Feedback regulation. Cell signaling networks often include feedback mechanisms that regulate the intensity and duration of signals. Positive feedback can amplify signals, while negative feedback can dampen or terminate signals. These feedback loops help maintain signal homeostasis and prevent overactivation or underactivation of cellular responses. Impairment of signaling mechanisms. Disruption or impairment of signaling mechanisms can have significant consequences for cellular function and contribute to the development of various diseases, including cancer, neurodegenerative disorders, metabolic diseases, and immune disorders. Such impairments can result from mutations in key components of signaling pathways, defects in receptor function, or dysregulation of upstream or downstream signaling molecules. Cancer. Many cancers are associated with the activation of oncogenic signaling pathways, such as the Ross-Raf-Meckert pathway, which promotes uncontrolled cell growth and survival. Mutations in receptor tyrosine kinases or downstream signaling molecules, such as PI3K or ACT, can result in persistent signaling that bypasses normal regulatory mechanisms, contributing to tumorigenesis. Neurodegenerative diseases. Disorders like Alzheimer's disease are associated with impaired signaling in pathways involved in cell survival, apoptosis, and synaptic plasticity. Dysfunctional receptor signaling can impair the communication between neurons, leading to cognitive decline and neurodegeneration. Metabolic disorders. Insulin resistance in type 2 diabetes results from impaired insulin signaling. In this case, desensitization of insulin receptors or defects in downstream signaling molecules, such as IRS, insulin receptor substrate, prevents proper glucose uptake and metabolism. Autoimmune diseases. In autoimmune disorders, the immune system fails to recognize self-antigens, often due to defects in signaling pathways that regulate immune cell activation and tolerance. This can result in the activation of T cells and the production of autoantibodies, leading to tissue damage. Role of phosphatases and kinases in signaling impairment. In addition to genetic mutations, the imbalance between kinases and phosphatases can contribute to signaling impairments. Hyperactive kinases and hypoactivation of phosphatases may lead to prolonged signaling and abnormal cellular behavior, whereas inactivation of kinases or overactive phosphatases may result in insufficient signaling and impaired cellular responses. MCQs. Which of the following mechanisms contributes to receptor desensitization? A. Phosphorylation of the receptor. B. Activation of intracellular second messenger. C. DNA binding by the receptor. D. Increased receptor synthesis. Which type of receptor is involved in fast, short-lived signaling events such as neurotransmission? A. G-beam coupled receptors. B. Ion channel receptors. C. Receptor tyrosine kinases. D. Intracellular receptors. Impairment of cell signaling mechanisms can lead to which of the following diseases? A. Cancer. B. Neurodegenerative diseases. C. Metabolic disorders. D. All of the above. MCQ answers. Answer. A phosphorylation of the receptor. Answer. B. Ion channel receptors. Answer. D. All of the above. This concludes the detailed explanation on receptor desensitization, cell signaling network, and impairment of signaling mechanisms. Let me know if you would like to proceed with other topics. You said. Cell signaling network, impairment of signaling mechanism. ChatGPT said. Cell signaling network and impairment of signaling mechanism. Cell signaling network. The cell signaling network is a complex and highly regulated system that allows cells to perceive and respond to external and internal stimuli. It involves multiple signaling pathways that interact and integrate signals to regulate cellular functions such as growth, differentiation, metabolism, immune response, and apoptosis. Signaling networks are composed of ligands, receptors, second messengers, kinases, phosphatases, transcription factors, and feedback mechanisms that ensure precise control of cellular activities. Components of the cell signaling network. Ligands signaling molecules. These include hormones, neurotransmitters, cytokines, and growth factors that initiate signaling by binding to receptors. Receptors. Receptors can be located on the cell surface, membrane receptors, or within the cell. Intracellular receptors. Examples include G-protein coupled receptors. GPCRs activate intracellular signaling via G-proteins. Receptor tyrosine kinases (RTKs) phosphorylate intracellular proteins to trigger downstream effects. Ligand-gated ion channels regulate ion flow in response to ligand binding. Intracellular receptors found in the cytoplasm or nucleus and activated by steroid hormones, thyroid hormones, and retinoic acid. Second messengers, small molecules that amplify and propagate the signal inside the cell. Examples include cyclic AMP GAMP activates protein kinase A (PKA) in GPCR signaling. Inositol triphosphate IP3 and diacylglycerol DAG mediate calcium release and protein kinase C (PKC) activation. Calcium Ca² regulates many cellular processes, including neurotransmission and muscle contraction. Effector proteins and transcription factors. These include kinases, phosphatases, and transcription factors that execute the cellular response. Examples: mitogen-activated protein kinases (MAPK) control cell proliferation and differentiation. Nuclear factor kappa B (NF-kappa B) regulates immune and inflammatory responses. Feedback mechanisms. Positive feedback amplifies the signal, e.g., calcium-induced calcium release in muscle contraction. Negative feedback suppresses the signal to prevent overstimulation, e.g., insulin signaling downregulation and diabetes. Major cell signaling pathways. GPCR signaling pathway. Binding activates G-protein, which stimulates secondary messengers like GAMP, IP3, and DAG. GPCRs regulate vision, olfaction, neurotransmission, and immune responses. Receptor tyrosine kinase (RTK) signaling. Growth factors like EGF, PDGF, and VEGF activate RTKs, leading to phosphorylation cascades. RTK signaling regulates cell proliferation, survival, and differentiation. WNT beta-catenin signaling pathway. Involved in embryogenesis, stem cell maintenance, and cancer development. WNT binding to frizzled receptors inhibits beta-catenin degradation, allowing it to enter the nucleus and activate transcription. Notch signaling pathway. Essential for cell fate determination, neurogenesis, and angiogenesis. Direct cell-cell communication where the Notch receptor is cleaved, releasing its intracellular domain to activate target genes. Toll-like receptor (TLR) signaling. Key in innate immunity, recognizing microbial patterns (PAMPs). Activation leads to NF-kappa B signaling and the production of inflammatory cytokines. PI3K-AKT pathway. Regulates cell survival, metabolism, and growth. Mutations in this pathway are implicated in cancer progression. JAK-STAT pathway. Activated by cytokines and growth factors. Plays a crucial role in immune function, hematopoiesis, and cell proliferation. Impairment of signaling mechanisms. Impairment or dysregulation of signaling pathways can lead to severe pathological conditions, including cancer, neurodegenerative diseases, metabolic disorders, and immune dysfunction. These impairments can result from mutations, post-translational modifications, receptor desensitization, or defective feedback mechanisms. Causes of impaired signaling: genetic mutations. Oncogenic mutations in RTKs, e.g., EGFR mutations in lung cancer, lead to uncontrolled cell proliferation. GPCR mutations can result in congenital night blindness, nephrogenic diabetes insipidus, and adrenal gland disorders. Mutations in tumor suppressor genes like P53 and PTEN lead to defective apoptosis and cancer development. Receptor dysfunction. Receptor downregulation due to excessive ligand exposure can result in conditions like insulin resistance. Loss of function mutations in TLRs can lead to immune suppression, increasing susceptibility to infections. Aberrant second messenger production. Overproduction of CAMP can lead to disorders like hyperthyroidism. Defective calcium signaling contributes to cardiovascular diseases and muscle dystrophy. Defective kinase and phosphatase regulation. Overactive kinases, e.g., hyperactivated MAPK pathway in cancer, promote uncontrolled cell growth. Impaired phosphatase activity, e.g., loss of PTEN function, leads to unregulated AKT signaling, promoting tumorigenesis. Neurodegenerative diseases. Defective neurotransmitter signaling leads to Alzheimer's, Parkinson's, and Huntington's disease. Impaired dopaminergic signaling in the basal ganglia contributes to Parkinson's disease. Immune system disorders. Overactive immune signaling, e.g., excessive TLR activation, leads to autoimmune diseases like rheumatoid arthritis and lupus. Defective JAK-STAT signaling results in immune deficiencies and hematopoietic disorders. Endocrine and metabolic disorders. Impaired insulin signaling leads to type 2 diabetes and metabolic syndrome. Thyroid hormone receptor mutations cause hypothyroidism and developmental abnormalities. Therapeutic approaches to correct signaling impairments: targeted therapies. Monoclonal antibodies (mAbs) used to block overactive receptors, e.g., trastuzumab for HER2-positive breast cancer. Tyrosine kinase inhibitors (TKIs) target hyperactivated kinases in cancer, e.g., imatinib for CML. JAK inhibitors, used for autoimmune disorders like rheumatoid arthritis. Gene therapy. Used to correct genetic defects in signaling pathways, e.g., CRISPR-based editing for cystic fibrosis. Small molecule inhibitors. Used to block aberrant signaling cascades, e.g., MEK inhibitors in melanoma. Immunotherapies. Checkpoint inhibitors PD-1, CTLA-4 inhibitors restore proper immune signaling in cancer patients. MCQs. Which of the following signaling pathways is directly involved in immune response regulation? A. WNT, beta-catenin pathway. B. MAPK pathway. C. Toll-like receptor pathway. D. Hedgehog pathway. Which defect in cell signaling is commonly associated with type 2 diabetes? A. Overactive GPCR signaling. B. Receptor tyrosine kinase downregulation. C. Overexpression of ion channels. D. Impaired WNT signaling. Mutations in which gene are commonly associated with tumor suppression failure and cancer development? A. ROS1 P53C, ACT, Notch. MCQ answers. Answer. C. Toll-like receptor pathway. Answer. B. Receptor tyrosine kinase downregulation. Answer. B. P53. This concludes the detailed discussion on cell signaling network and impairment of signaling mechanisms. Let me know the next topic. Search. Reason. DNA replication, repair, and recombination. DNA replication, repair, and recombination are essential processes ensuring the stability and integrity of the genetic material in all living organisms. These mechanisms allow cells to accurately duplicate their genomes, correct errors, and facilitate genetic diversity. 1. DNA replication 1.1. Overview. DNA replication is a semi-conservative process in which each parental DNA strand serves as a template for the synthesis of a new complementary strand. This process occurs in the S phase of the cell cycle and ensures the accurate transmission of genetic information. 1.2. Steps of DNA replication 1.2.1. Initiation. Origin of replication or DNA replication begins at specific sequences called origins of replication. Initiator proteins. In prokaryotes, DNA A binds to OREC, initiating strand separation. In eukaryotes, the origin recognition complex ORC recruits helicase. Helicase DNA B in prokaryotes, MCM in eukaryotes unwinds the DNA double helix. Single-strand binding proteins SSBs prevent reannealing of separated strands. Topoisomerase relieves supercoiling stress ahead of the replication fork. 1.2.2. Elongation. Primase synthesizes short RNA primers to provide a 3' group for DNA polymerase. DNA polymerase. Prokaryotes, DNA polymerase 3 is the primary enzyme for elongation. Eukaryotes, DNA polymerase alpha primase activity delta lagging strand synthesis and epsilon leading strand synthesis. Leading strand synthesis, continuous synthesis in the 5' right pointing arrow 3 direction. Lagging strand synthesis, discontinuous synthesis through Okazaki fragments, later joined by DNA ligase. Sliding clamp beta clamp in prokaryotes, PCNA in eukaryotes enhances polymerase processivity. 1.2.3. Termination. Prokaryotes, replication ends at tear sequences, recognized by the 2' protein, preventing helicase movement. Eukaryotes, ends of linear chromosomes require telomerase, a reverse transcriptase that extends telomeres to prevent loss of genetic material. 2. DNA repair mechanisms. DNA is constantly subjected to damage from replication errors, chemical modifications, and external factors UV, radiation. Cells have evolved repair mechanisms to maintain genome integrity. 2.1. Direct repair. Photoreactivation. UV-induced thymine dimers are repaired by DNA photolysis prokaryotes and some eukaryotes. 6-methylguanine DNA methyltransferase MGMT repairs alkylated guanine. 2.2. Excision repair. Base excision repair BER. Repairs small, non-helix distorting base modifications e.g., deaminated cytosine right pointing arrow uracil. DNA glycosylase removes the damaged base, AP endonuclease cuts the backbone, and DNA polymerase beta eukaryotes or I prokaryotes fills the gap. Nucleotide excision repair NER. Repairs bulky lesions e.g., pyrimidine dimers, chemical adducts. UVRABC endonuclease prokaryotes and XPC, XPA, XPB, XPD eukaryotes recognize and excise damaged nucleotides. DNA polymerase fills the gap. 2.3. Mismatch repair MMR. Corrects replication errors, mismatched bases, small indels. Prokaryotes, MUTS recognition, MUDL recruitment and MUF endonuclease. Eukaryotes, MLH1, MSH2, PMS2. Mutations in MMR genes lead to Lynch syndrome, hereditary non-polyposis colorectal cancer, HNPCC. 2.4. Double-strand break DSB repair. Homologous recombination HR error-free repair using a homologous sequence as a template. Uses BRCA1, BRCA2, RAD51 in eukaryotes. Non-homologous end joining NHEJ error-prone repair that directly ligates broken ends. Uses Q70, Q80, DNA PKCs. 2.5. Translesion synthesis TLS. Specialized polymerases e.g., DNA pol eta, zeta bypass DNA lesions at the cost of introducing mutations. 3. DNA recombination. 3.1. Homologous recombination HR. Occurs during meiosis, mitosis DSB repair, and horizontal gene transfer. Key steps. Strand invasion, RECA prokaryotes, RAD51 eukaryotes. Holiday junction formation and resolution. 3.2. Site-specific recombination mediated by recombinases e.g., Cre, lambda integrase. Example, integration of bacteriophage lambda into the E. coli genome. 3.3. Transposition. Transposable elements, jumping genes, move within genomes via transposase. Types. Class 1 retrotransposons, RNA intermediates, e.g., lines, signs. Class 2 DNA transposons, direct DNA movement, e.g., is elements, P elements. Practice questions with answers. Previous year question, Gate Ecology and Evolution Q1, which of the following is the most error-prone DNA repair mechanism? A. Homologous recombination. B. Base excision repair. C. Non-homologous end joining. D. Nucleotide excision repair. Answer. C. Non-homologous end joining. NHEJ directly joins broken DNA ends without a template, often introducing mutations. Multiple choice questions MCQs. Q2. Which enzyme is responsible for removing RNA primers in eukaryotic DNA replication? A. DNA pol 3. B. DNA pol alpha. C. DNA pol delta. D. RNase H. Answer. D. RNase H. RNase H removes RNA primers, and DNA pol delta fills the gaps. Q3. Which protein is primarily responsible for strand invasion during homologous recombination in eukaryotes? A. RECA. Beat Rad 51C BRCA1D mutts. Answer. Beat Rad 51. Rad 51 facilitates strand invasion by pairing homologous DNA sequences. This detailed coverage of DNA replication, repair, and recombination aligns with PhD entrance preparation standards and includes previous year questions and MCQs for effective revision. Let me know if you need further elaboration. You said. Unit of replication, enzymes involved, replication origin and replication fork, fidelity of replication, ChatGPT said. DNA replication, unit of replication, enzymes, replication origin, replication fork, and fidelity. DNA replication is a highly regulated process ensuring the accurate duplication of genetic material. This section covers key aspects such as the unit of replication, enzymes involved, replication origin, replication fork, and the fidelity of replication. 1. Unit of replication. The unit of replication, or replicon, is the segment of DNA that is replicated from a single origin of replication. Prokaryotes, e.g., E. coli, have a single replicon due to a single origin of replication, or EC, on their circular chromosome. Eukaryotes, e.g., humans, have multiple replicons with multiple origins of replication to accommodate large linear chromosomes. Each replicon functions independently, ensuring the entire genome is replicated efficiently. 2. Enzymes involved in DNA replication. 2.1. Helicase unwinds the DNA double helix by breaking hydrogen bonds between base pairs. Prokaryotes, DNAB, part of the chromosome complex. Eukaryotes, minichromosome maintenance, MCM complex, MCM 2-7. 2.2. Single-strand binding proteins, SSBs, prevent reannealing of separated DNA strands. Prokaryotes, SSB protein. Eukaryotes, replication protein A, RPA. 2.3. Topoisomerase relieves supercoiling caused by helicase activity. Topoisomerase I introduces single-strand breaks. Topoisomerase 2, e.g., DNA gyrase in prokaryotes introduces double-strand breaks to relieve torsional strain. 2.4. Primase synthesizes RNA primers to provide a free 3-keto group for DNA polymerase. Prokaryotes, DNAG primase. Eukaryotes, DNA polymerase alpha has primase activity. 2.5. DNA polymerases. Prokaryotic DNA polymerases. DNA pol I removes RNA primers and fills gaps. DNA pol 3, main polymerase for elongation. DNA pol 2, IV, involved in DNA repair. Eukaryotic DNA polymerases. DNA pol alpha initiates replication by adding RNA primers. DNA pol delta synthesizes the lagging strand. DNA pol epsilon synthesizes the leading strand. DNA pol beta involved in DNA repair. 2.6. Sliding clamp enhances polymerase processivity, ability to add nucleotides continuously. Prokaryotes, beta clamp part of the DNA pol 3 holoenzyme. Eukaryotes, proliferating cell nuclear antigen, pCNA. 2.7. Clamp loader loads the sliding clamp onto DNA. Prokaryotes, gamma complex part of DNA pol 3 holoenzyme. Eukaryotes, RFC, replication factor C. 2.8. RNase H and DNA ligase RNase H removes RNA primers in eukaryotes. DNA ligase seals nicks in DNA by forming phosphodiester bonds. Prokaryotic DNA ligase uses NAD+. Eukaryotic DNA ligase uses ATP. 3. Replication origin and replication fork. 3.1. Replication origin. The origin of replication, ORI, is the specific DNA sequence where replication begins. Prokaryotes. Single origin, ORI-C in E. coli. ORI-C contains DNA A boxes where DNA A binds to initiate replication. Eukaryotes. Multiple origins, ARS, autonomous replication sequences in yeast. Origin recognition complex, ORC, binds to origins and recruits replication proteins. 3.2. Replication fork. The replication fork is a Y-shaped structure where active DNA synthesis occurs. Key components of the replication fork. Leading strand, synthesized continuously in the 5 feet right pointing arrow 3 direction. Lagging strand, synthesized discontinuously as Okazaki fragments. Replication proteins, helicase, primase, DNA polymerases, etc. Function at the fork. Bidirectional replication, DNA synthesis proceeds in both directions from the origin. 4. Fidelity of DNA replication. DNA replication is highly accurate, with an error rate of 1 in 10 to the power of 9 10 nucleotides due to multiple proofreading and repair mechanisms. 4.1. Proofreading by DNA polymerase. DNA polymerases have 3 feet right pointing arrow 5, exonuclease activity, allowing them to remove incorrect nucleotides. Prokaryotes, DNA pol 3 has proofreading ability. Eukaryotes, DNA pol delta and epsilon have proofreading functions. 4.2. Mismatch repair, MMR, system corrects errors missed by DNA polymerase. Prokaryotes, MUTS detects mismatch, MUDL, and MUTH cleaves incorrect strand. Eukaryotes, MSH2, MLH1, PMS2. 4.3. DNA damage repair mechanisms. Base excision repair, BER, corrects minor base modifications. Nucleotide excision repair, NER, removes bulky DNA lesions. Double strand break, DSB, repair, homologous recombination, HR, error-free. Non-homologous end joining, NHEJ, error-prone. 4.4. Role of telomerase in replication. Fidelity prevents loss of genetic information at chromosome ends. Eukaryotic linear chromosomes require telomerase to extend telomeres. Practice questions with answers. Previous year question, gate ecology and evolution. Q1. Which of the following DNA polymerases is responsible for leading strand synthesis in eukaryotes? A. DNA polymerase alpha. B. DNA polymerase delta. C. DNA polymerase epsilon. D. DNA polymerase 3. Answer. C. DNA polymerase epsilon. DNA pol epsilon synthesizes the leading strand, while DNA pol delta synthesizes the lagging strand. Multiple choice questions. MCQs. Q2. What is the function of DNA helicase in replication? A. Synthesizing RNA primers. B. Relieving supercoiling. C. Unwinding the DNA double helix. D. Proofreading the newly synthesized strand. Answer. C. Unwinding the DNA double helix. Helicase breaks hydrogen bonds between base pairs to separate the strands. Q3. Which of the following is not a function of DNA ligase? A. Sealing nicks between Okazaki fragments. B. Adding nucleotides to the leading strand. C. Forming phosphodiester bonds. D. Connecting repaired DNA segments. Answer, B. Adding nucleotides to the leading strand. DNA ligase does not synthesize DNA, it seals nicks. This detailed explanation covers all major aspects of DNA replication while maintaining a high academic standard for PhD entrance preparation. Let me know if you need further elaboration. You said. Fidelity of replication, extrachromosomal replicons, DNA damage and repair mechanisms, homologous and site-specific recombination, ChatGPT said. DNA replication fidelity, extrachromosomal replicons, DNA damage and repair, and recombination. DNA replication, repair, and recombination are essential processes that maintain genome integrity, prevent mutations, and enable genetic diversity. Below is a detailed explanation of these topics. 1. Fidelity of DNA replication. The fidelity of DNA replication ensures the accurate duplication of genetic material, minimizing mutations. The error rate of replication is approximately 1 error per 10 to the power of 9 10 nucleotides due to several proofreading and repair mechanisms. 1.1. Mechanisms ensuring high fidelity. 1.1.1. Base pair complementarity. DNA polymerases follow Watson-Crick base pairing rules, AT, GC, ensuring accurate incorporation of nucleotides. 1.1.2. Proofreading by DNA polymerases. DNA polymerases have 3 feet right pointing arrow 5, exonuclease activity, allowing them to remove incorrectly incorporated nucleotides. Prokaryotes, DNA pol 3 has proofreading ability. Eukaryotes, DNA pol delta and epsilon have proofreading functions. 1.1.3. Mismatch repair MMR corrects replication errors missed by proofreading. Prokaryotes, MUTS detects mismatches, MIDAL recruits smooth, which cuts the incorrect strand. Eukaryotes, MSH2, MLH1, PMS2 perform similar functions. 1.1.4. DNA damage repair mechanisms. Base excision repair BER corrects minor lesions. Nucleotide excision repair NER removes bulky DNA adducts. Double strand break DSB repair ensures genome stability. 2. Extrachromosomal replicons. Extrachromosomal replicons are DNA elements that replicate independently of chromosomal DNA. These include plasmids, episomes, viral genomes, and mitochondrial DNA. 2.1. Types of extrachromosomal replicons. 2.1.1. Plasmids. Small, circular DNA molecules found in bacteria and some eukaryotes. Carry non-essential genes, including those for antibiotic resistance, are plasmids or virulence factors. Replicate independently via the theta replication or rolling circle replication mechanism. 2.1.2. Episomes. Plasmids that can integrate into the host genome, e.g., F plasmid and E. coli. Facilitate horizontal gene transfer via conjugation. 2.1.3. Viral genomes. Some viruses maintain extrachromosomal replicons inside host cells, e.g., HIV provirus. Bacteriophage lambda integrates into bacterial DNA as a prophage during lysogeny. 2.1.4. Mitochondrial and chloroplast DNA circular genomes inherited maternally in eukaryotes. Replicate using a replicative DNA polymerase, e.g., POLG in humans. 3. DNA damage and repair mechanisms. DNA is constantly exposed to spontaneous and induced damage, leading to mutations. Cells have evolved repair mechanisms to counteract these errors. 3.1. Types of DNA damage. 3.1.1. Spontaneous damage. Depurination, loss of purine bases A, G. Deamination, cytosine converts to uracil. Tautomeric shifts, base misfiring due to altered hydrogen bonding. 3.1.2. Induced damage. UV radiation, forms pyrimidine dimers, TT or CC dimers. Ionizing radiation, X-rays, gamma rays causes double-strand breaks, DSBs. Chemical mutagens, alkylating agents, oxidative damage. 3.2. DNA repair mechanisms. 3.2.1. Direct repair. Photoreactivation, DNA photolysis repairs pyrimidine dimers in prokaryotes. Alkyltransferases, e.g., MGMT, remove alkyl groups from guanine. 3.2.2. Excision repair. Base excision repair, BER, repairs single base modifications. DNA glycosylase removes the altered base. AP endonuclease cuts the DNA backbone. DNA polymerase beta eukaryotes or I prokaryotes fills the gap. Nucleotide excision repair, NER, repairs bulky lesions. Prokaryotes, UVR ABC endonuclease. Eukaryotes, XPC, XPA, XPB, XPD recognize damage and excise it. 3.2.3. Mismatch repair, MMR. Recognizes and fixes replication errors. MUTS MIDAL prokaryotes and MSH2 MLH1 PMS2 eukaryotes. 3.2.4. Double strand break DSB repair homologous recombination HR error-free, uses sister chromatid as a template. Non-homologous end joining NHEJ error-prone, directly joins broken ends. 4. Homologous and site-specific recombination. Recombination is the exchange of genetic material between DNA molecules and plays a crucial role in genetic diversity, repair, and evolution. 4.1. Homologous recombination HR. Occurs during meiosis, mitosis DSB repair, and horizontal gene transfer. Requires long sequence homology. Key steps. Strand invasion. RACUP prokaryotes or RAD51 eukaryotes binds to the single-stranded DNA and searches for homologous sequences. Holiday junction formation. DNA strands exchange between homologous chromosomes. Branch migration. The junction moves along the DNA. Resolution. Holiday junctions are cleaved by resolvases. Can result in crossover genetic exchange or non-crossover gene conversion. Examples. Meiotic recombination crossing over. DSB repair by HR BRCA1 and BRCA2 in eukaryotes. 4.2. Site-specific recombination. Recombination occurs at specific DNA sequences, mediated by recombinases. Does not require long sequence homology. Key enzymes. Integrases e.g., lambda integrase in bacteriophages. Cre-recombinase used in genetic engineering. Transposases for transposon movement. Examples. Integration of bacteriophage lambda into E. coli genome. VDJ recombination in immunoglobulin genes. Cre-LoxP system for conditional gene knockout. Practice questions with answers. Previous year question. Gate ecology and evolution. Q1. Which DNA repair mechanism is responsible for fixing UV-induced thymine dimers? A. Base excision repair. B. Nucleotide excision repair. C. Non-homologous end joining. D. Mismatch repair. Answer. B. Nucleotide excision repair. NER removes bulky DNA lesions such as thymine dimers. Multiple choice questions. MCQs. Q2. What is the function of RECA in homologous recombination? A. DNA polymerization. B. Recognition of mismatch repair sites. C. Strand invasion and homologous pairing. D. Cleaving phosphodiester bonds. Answer: C. Strand invasion and homologous pairing. Q3. Which enzyme is responsible for resolving holiday junctions? A. DNA polymerase. B. Ligase. C. Resolvase. D. Topoisomerase. Answer: C. Resolvase. This high-level PhD entrance preparation note comprehensively covers replication fidelity, extrachromosomal replicons, DNA repair, and recombination mechanisms with previous year questions and MCQs for effective revision. Let me know if you need further clarification. You said. RNA synthesis and processing, transcription factors and machinery. ChatGPT said. RNA synthesis and processing, transcription factors and machinery. RNA synthesis, or transcription, is the process by which genetic information from DNA is copied into RNA by RNA polymerase. In eukaryotes and prokaryotes, transcription involves various transcription factors, regulatory elements, and enzymes that ensure accurate gene expression. 1. Overview of transcription. Transcription occurs in 3 phases. Initiation, RNA polymerase binds to the promoter region with the help of transcription factors. Elongation, RNA polymerase synthesizes RNA by complementary base pairing with the DNA template strand. Termination, transcription stops, and the RNA molecule is released. In prokaryotes, transcription occurs in the cytoplasm, while in eukaryotes, it takes place in the nucleus before RNA is processed and exported to the cytoplasm. 2. Transcription machinery in prokaryotes. 2.1. RNA polymerase in prokaryotes. Prokaryotes have a single RNA polymerase enzyme that transcribes all types of RNA, mRNA, rRNA, and tRNA. This enzyme consists of core enzyme alpha BB, omega responsible for RNA synthesis. Sigma factor sigma recognizes the promoter and initiates transcription. 2.2. Transcription factors in prokaryotes. Sigma factors different sigma factors recognize different promoters. Example: sigma to the power of 7 housekeeping genes, sigma cubed squared heat shock response. Repressors and activators. Lac repressor LAC inhibits transcription of the lac operon in E. coli. CAMP receptor protein CRP enhances transcription when bound to CAMP. 2.3. Promoter elements in prokaryotes. 10 region Pribnow box, TATAAT, and -35 region TTGACA help RNA polymerase bind and initiate transcription. 2.4. Termination in prokaryotes. Rho-dependent termination requires the Rho factor, which dissociates RNA from DNA. Rho-independent termination involves a GC-rich hairpin loop followed by a poly-U sequence. 3. Transcription machinery in eukaryotes. 3.1. RNA polymerases in eukaryotes. Eukaryotes have 3 major RNA polymerases. RNA polymerase I right pointing arrow synthesizes rRNA, 28S, 18S, 5.8S rRNAs. RNA polymerase 2 right pointing arrow transcribes mRNA and some snRNA. RNA polymerase 3 right pointing arrow transcribes tRNA, 5S rRNA, and other small RNAs. 3.2. General transcription factors GTFs in eukaryotes. RNA polymerase 2 requires several general transcription factors to initiate transcription. Factor function. TFIID contains TBP, binds to TATA box and recruits other factors. TFIIB helps RNA pol 2 position at the promoter. TFIIF stabilizes RNA pol 2 binding. TFIIE recruits TFIIH. TFIIH helicase activity unwinds DNA and kinase activity phosphorylates RNA pol 2 CTD. 3.3. Eukaryotic promoter elements. Core promoter. TATA box -25 region found in some genes, binds TFIID. Initiator INR, sequence, alternative to TATA box. Breed TFIIB recognition element, located upstream of TATA. DPE, downstream promoter element found in TATA-less promoters. Enhancers and silencers. Enhancers, increase transcription by binding activators. Silencers, decrease transcription by binding repressors. 3.4. Termination in eukaryotes. RNA polymerase 2 termination involves cleavage of the RNA transcript and polyadenylation. RNA polymerase I and 3 terminate via different mechanisms involving specific termination signals. 4. RNA processing in eukaryotes. After transcription, primary RNA transcripts undergo several modifications to become mature RNA. 4.1. mRNA processing in eukaryotes. 5' capping. Addition of a 7-methylguanosine M to the power of 7G cap at the 5' end. Helps with mRNA stability and ribosome recognition. Splicing. Removal of introns non-coding regions and joining of exons coding regions. Spliceosome complex SNRNPs, U1, U2, U4, U5, U6 facilitates splicing. Polyadenylation. Addition of a poly-A tail approximately 200 adenines at the 3' end. Protects mRNA from degradation and aids translation. RNA editing. Some mRNAs undergo base modifications, e.g., 8I editing adenosine to inosine in neurotransmitter receptors. 4.2. rRNA and tRNA processing. rRNA processing. Precursor 45S rRNA is cleaved into 18S, 28S, and 5.8S rRNAs. tRNA processing. Trimming of 5' and 3' ends. Addition of CCA sequence at 3' end. Base modifications e.g., pseudouridine, inosine. 5. Regulation of transcription. 5.1. Epigenetic regulation. Histone modifications. Acetylation activates transcription. Methylation can activate or repress. DNA methylation. CpG island methylation silences genes. 5.2. Transcriptional activators and repressors. Activators bind enhancers to promote transcription. Repressors bind silencers to inhibit transcription. 5.3. Chromatin remodeling complexes. SWI SNF complex opens chromatin for transcription. Polygome group proteins silence genes. Practice questions with answers. Previous year question. Gate ecology and evolution. Q1. Which of the following is a function of the TATA binding protein TBP? A. Termination of transcription. B. RNA splicing. C. Recognition of the TATA box in promoters. D. Translation initiation. Answer. C. Recognition of the TATA box in promoters. Multiple choice questions. MCQs. Q2. Which RNA polymerase is responsible for synthesizing tRNA in eukaryotes? A. RNA polymerase I. B. RNA polymerase 2. C. RNA polymerase 3. D. RNA polymerase IV. Answer. C. RNA polymerase 3. Q3. Which transcription factor in eukaryotes is responsible for unwinding DNA at the promoter? A. TFIIB. B. TFIIEC, TFIIHD, TFIID. Answer: C. TFIIH. This detailed PhD entrance preparation note covers transcription factors, RNA synthesis machinery, and RNA processing with previous year questions and MCQs for revision. Let me know if you need any further clarifications. You said, formation of initiation complex, transcription activator and repressor, RNA polymerases, capping elongation, and termination. ChatGPT said, formation of initiation complex, transcription activators and repressors, RNA polymerases, capping, elongation, and termination 1. Formation of the initiation complex. 1.1. Prokaryotic transcription initiation complex in prokaryotes, RNA polymerase directly binds to the promoter with the help of sigma factors. Steps. RNA polymerase holoenzyme formation. The core enzyme alpha BB omega associates with a sigma factor to form the holoenzyme. Promoter recognition. The sigma factor recognizes the -10 Pribnow box, TATAAT, and -35 TTGAC8 sequences. Closed complex formation. The RNA polymerase holoenzyme binds to the promoter but the DNA remains double-stranded. Open complex formation. RNA polymerase unwinds approximately 12 to 14 pp of DNA near the transcription start site +1. Abortive initiation. Short RNA sequences, approximately 10 nucleotides, are synthesized and released. Promoter clearance. Once RNA synthesis stabilizes, approximately 10 nucleotides long, sigma factor dissociates, and RNA polymerase transitions to elongation. 1.2. Eukaryotic transcription initiation complex. In eukaryotes, transcription initiation is more complex, requiring multiple general transcription factors, GTFs. Steps. Pre-initiation complex formation. TFIID containing TBP, tau binding protein, binds to the tau box -25 region. TFIIB recognizes the Br element and helps recruit RNA polymerase 2. TFIIF stabilizes RNA pol 2 binding to the promoter. TFIIE and TFIIH with helicase and kinase activity are recruited. DNA unwinding. TFIIH helicase activity unwinds DNA, forming the open complex. TFIIH kinase activity phosphorylates the C-terminal domain, CTD, of RNA pol 2, triggering promoter clearance. Promoter clearance and transition to elongation. RNA pol 2 detaches from general transcription factors and starts elongation. 2. Transcription activators and repressors 2.1. Transcription activators. Definition: proteins that enhance transcription by binding to enhancers or interacting with transcription machinery. Mechanism of action. Recruit RNA polymerase and transcription factors. Open chromatin by recruiting histone acetyltransferases HATs. Example: CAM receptor protein CRP in prokaryotes. 2.2. Transcription repressors. Definition: proteins that inhibit transcription by blocking activator binding or compacting chromatin. Mechanism of action. Bind to silencers and prevent RNA polymerase binding. Recruit histone deacetylases HDACs to close chromatin. Example: lac repressor LAC in E. coli. 3. RNA polymerases. 3.1. Prokaryotic RNA polymerase. A single RNA polymerase transcribes all RNA types. Composed of a core enzyme alpha BB, omega, and a sigma factor for promoter recognition. 3.2. Eukaryotic RNA polymerases. RNA polymerase I transcribes rRNA 28S, 18S, 5.8S rRNA. RNA polymerase II transcribes mRNA, snRNA, mRNA. RNA polymerase III transcribes tRNA, 5S rRNA, and small RNAs. Each polymerase recognizes distinct promoter elements and requires specific transcription factors. 4. Capping of mRNA, eukaryotic only function. Protects mRNA from degradation. Enhances translation initiation process. Addition of a 7-methylguanosine M to the power of 7G cap at the 5' end. Enzymes involved. RNA triphosphatase removes the gamma phosphate. Ganylyltransferase adds GMP in a 5' triphosphate bond. Methyltransferase adds a methyl group to guanine. Prokaryotic mRNA lacks capping and is rapidly degraded after use. 5. Elongation of transcription 5.1. Prokaryotic elongation. After sigma factor dissociation, the core enzyme moves along the DNA, adding ribonucleotides complementary to the template strand. RNA polymerase maintains a 17 BP transcription bubble. 5.2. Eukaryotic elongation. RNA pol 2 continues transcription while associated with elongation factors. CTD phosphorylation by TFIIH kinase allows recruitment of RNA processing factors, splicing, capping. 6. Termination of transcription. 6.1. Prokaryotic termination. 2 main mechanisms. Rho-dependent termination. Rho factor, an RNA helicase, binds to a site on RNA and disrupts RNA-DNA binding. Rho-independent termination. Formation of a GC-rich hairpin loop followed by a poly-U stretch leads to RNA detachment. 6.2. Eukaryotic termination. RNA polymerase I uses termination factor TTF1 to stop transcription. RNA polymerase III stops transcription at specific poly-T sequences. RNA polymerase II. Cleavage of the transcript at the polyadenylation signal AAU-AAA. Addition of a poly-A tail by poly-A polymerase for stability. Practice questions with answers. Previous year question: Gait ecology and evolution. Q1. Which of the following transcription factors is responsible for unwinding DNA during transcription initiation in eukaryotes? A. TFIID. B. TFIIEC, TFIIHD, TFIIB. Answer: C. TFIIH. Multiple choice questions MCQs. Q2. What is the role of the 7-methylguanosine cap in eukaryotic mRNA? A. Prevents ribosome binding. B. Facilitates mRNA degradation. C. Enhances translation and stability. D. Blocks RNA polymerase binding. Answer: C. Enhances translation and stability. Q3. Which enzyme is responsible for adding the 5' cap to eukaryotic mRNA? A. RNA polymerase I. B. RNA polymerase II. C. Ganylyltransferase. D. Poly-A polymerase. Answer: C. Ganylyltransferase. This detailed PhD entrance study note covers transcription initiation, RNA polymerases, activators, repressors, elongation, capping, and termination with previous year and practice questions. Let me know if you need more explanations or specific topics. You said RNA processing, RNA editing, splicing. and polyadenylation, structure, and function of different types of RNA, RNA transport. ChatGPT said. RNA processing, editing, splicing, polyadenylation, structure and function of different RNAs, and RNA transport 1. RNA processing. RNA processing involves modifications to the primary RNA transcript to produce functional RNA molecules. These modifications include capping, splicing, polyadenylation, and editing. Prokaryotes, mRNA is mostly unprocessed and translated immediately. Eukaryotes, pre-mRNA undergoes extensive processing in the nucleus before being transported to the cytoplasm for translation. 2. RNA editing. RNA editing alters the nucleotide sequence of RNA after transcription. This process can generate different protein isoforms from a single gene. 2.1. Types of RNA editing. Substitution editing. Adenosine to inosine ADI editing, catalyzed by adenosine deaminases acting on RNA ADARs. Alters codon specificity, affecting protein function. Cytosine to uracil CDU editing, catalyzed by cytidine deaminases. Example, apolipoprotein B, ApoB mRNA editing in humans, which produces 2 different proteins. Insertion deletion editing. Mediated by guide RNAs GRNAs, common in mitochondrial RNA of trypanosomes. 3. RNA splicing. RNA splicing removes non-coding introns and joins exons to produce a mature mRNA transcript. 3.1. Types of splicing. Spliceosome-mediated splicing, pre-mRNA splicing. Occurs in eukaryotic nuclei and involves small nuclear RNAs, snRNAs, forming a spliceosome complex. Steps of splicing. Recognition of the 5' splice site GU and 3' splice site AG. Formation of a lariat structure at the branch point, a residue. Exon ligation and intron removal. Self-splicing, ribozyme-mediated splicing. Group 1 introns, require GTP as a cofactor. Group 2 introns, form a lariat similar to spliceosomal splicing. Alternative splicing. Generates multiple protein isoforms from a single gene. Examples. Fibronectin gene, different splice variants function in different tissues. Drosophila descan gene, generates thousands of protein isoforms. 4. Polyadenylation. Polyadenylation adds a poly A tail to the 3' end of eukaryotic mRNA to enhance stability and translation. 4.1. Process of polyadenylation cleavage at the polyadenylation signal AAU-AAA. Addition of approximately 200 adenine residues by poly A polymerase PAP. Binding of poly A binding proteins PABPs for stability and translation efficiency. 4.2. Functions of the poly A tail. Enhances mRNA stability, prevents degradation. Aids in nuclear export. Facilitates translation initiation by interacting with the 5' cap. 5. Structure and function of different types of RNA. 5.1. Messenger RNA, mRNA. Structure, linear, single-stranded with a 5' cap and 3' poly A tail, eukaryotes only. Function, carries genetic information from DNA to ribosomes for translation. Prokaryotic versus eukaryotic mRNA. Prokaryotic mRNA, polycistronic, encodes multiple proteins. Eukaryotic mRNA, monocistronic, 1 gene per mRNA. 5.2. Ribosomal RNA, rRNA. Structure, forms complex secondary structures in ribosomes. Function, catalyzes peptide bond formation during translation, ribozyme activity. Types. Prokaryotic rRNA, 16S small subunit, 23S, 5S large subunit. Eukaryotic rRNA, 18S small subunit, 28S, 5.8S, 5S large subunit. 5.3. Transfer RNA, tRNA. Structure, cloverleaf shape with anticodon and amino acid attachment site. Function, transfers amino acids to ribosomes during translation. Post-transcriptional modifications, pseudouridine, inosine. 5.4. Small nuclear RNA, snRNA. Function, forms the spliceosome, involved in RNA splicing. Examples, U1, U2, U4, U5, U6 snRNAs. 5.5. Small nucleolar RNA, snRNA. Function, guides rRNA modifications, methylation and pseudourylation. Example, CT-box snRNAs modify ribosomal RNA. 5.6. MicroRNA, mRNA, and small interfering RNA, siRNA, mRNA, regulates gene expression by blocking translation. siRNA, degrades specific mRNAs via RNA interference RNAi. 6. RNA transport. After processing, RNA must be transported to specific cellular locations for function. 6.1. Nuclear export of mRNA mediated by nuclear pore complexes, NPCs. Requires export factors, e.g., NXF1 NXT1 complex in mammals. Export is coupled with splicing and polyadenylation. 6.2. tRNA and rRNA transport. tRNA, processed in the nucleus and exported by exportin T. rRNA, assembled into ribosomal subunits in the nucleolus, then exported to the cytoplasm. Practice questions with answers. Previous year question, Gate Ecology and Evolution, Q1. Which RNA processing event involves the removal of introns and joining of exons? A. Capping. B. Splicing. C. Polyadenylation. D. RNA editing. Answer, B. Splicing. Multiple choice questions, MCQs. Q2. Which enzyme catalyzes the addition of the 3' poly A tail in eukaryotic mRNA? A. RNA polymerase 2. B. Guanylyltransferase. C. Poly A polymerase D. ADAR. Answer, C. Poly A polymerase. Q3. Which RNA molecule has catalytic activity in the ribosome? A. tRNA. B. mRNA. C. rRNA. D. snRNA. Answer, C. rRNA. This detailed PhD entrance study note covers RNA processing, RNA editing, splicing, polyadenylation, RNA structure and function, and RNA transport, along with previous year and practice questions. Let me know if you need any further clarifications. You said. Protein synthesis and processing, ribosome, formation of initiation complex, initiation factors and their regulation, elongation and elongation factors. ChatGPT said. Protein synthesis and processing 1. Ribosome, structure and function 1.1. Ribosome composition. Ribosomes are macromolecular machines that translate mRNA into proteins. They consist of ribosomal RNA, rRNA, and ribosomal proteins. Prokaryotic ribosome, 70S, small subunit 30S, 16S rRNA plus proteins, large subunit 50S, 23S and 5S rRNA plus proteins, eukaryotic ribosome, 80S, small subunit 40S, 18S rRNA plus proteins, large subunit 60S, 28S, 5.8S, and 5S rRNA plus proteins, 1.2. Ribosome function. The ribosome decodes mRNA and catalyzes peptide bond formation. Key sites. A site aminoacyl site binds incoming tRNA with an amino acid. P site peptidyl site holds the growing polypeptide chain. E site exit site where uncharged tRNA exits the ribosome. 2. Formation of the initiation complex 2.1. Prokaryotic initiation complex Shine-Dalgarno sequence. Located upstream of the start codon August. Recognized by 16S rRNA of the 30S subunit. Steps. 30S ribosomal subunit binding. Initiation factor 3 IF3 prevents premature 50S binding. Initiation factor 1 IF1 blocks the A site. mRNA and FMET tRNA binding. IF2 GTP binds FMET tRNA FMET to the P site. 50S subunit assembly. GTP hydrolysis releases If1, IF2, and IF3. Functional 70S ribosome is assembled. 2.2. Eukaryotic initiation complex cosec sequence. Helps position the ribosome with the correct August start codon. Steps. 40S subunit binds initiation factors EIFs, EIF1, EIF1A, and EIF3 keep the 40S subunit dissociated from 60S. EIF2 GTP binds MET tRNA MET and brings it to the P site. mRNA recognition and scanning. EIF4 complex EIF4E, EIF4G, EIF4A, EIF4B binds the 5' cap and unwinds secondary structures. The 40S subunit scans for the August start codon. 60S subunit joining. EIF5B GTP hydrolysis triggers release of EIFs. Functional EDS ribosome is assembled. 3. Initiation factors and the regulation 3.1. Prokaryotic initiation factors If1 blocks the A site until the ribosome is ready for elongation. IF2 GTP binding protein that helps FMET tRNA bind to the ribosome. If3 prevents premature binding of the 50S subunit. 3.2. Eukaryotic initiation factors EIF2 brings MET tRNA to the ribosome. EIF4E binds the 5' cap of mRNA. EIF4G scaffolding protein that bridges EIF4E and other factors. EIF5B facilitates 60S subunit joining. Regulation. Phosphorylation of EIF2 alpha inhibits translation during stress conditions. mTOR signaling pathway regulates EIF4E availability, controlling protein synthesis rates. 4. Elongation of translation. 4.1. Steps of elongation aminoacyl tRNA entry. Elongation factor 2 EF2 GTP binds aminoacyl tRNA and delivers it to the A site. GTP hydrolysis releases EF2. Peptide bond formation. Peptidyltransferase 23 srRNA in prokaryotes, 28 srRNA in eukaryotes catalyzes bond formation. Peptide chain moves from P site tRNA to A site tRNA. Translocation. Elongation factor G EFG prokaryotes EF2 eukaryotes hydrolyzes GTP to shift the ribosome forward by one codon. The uncharged tRNA moves to the E site and exits. 5. Elongation factors in the regulation 5.1. Prokaryotic elongation factors EF2 brings aminoacyl tRNA to the A site. EFTs regenerates EF2 GTP. EFG promotes ribosome translocation. 5.2. Eukaryotic elongation factors EF1A analogous to EF2 delivers aminoacyl tRNA. EF1B regenerates EF1A GTP. EF2 promotes ribosome translocation. Regulation. Diphtheria toxin and ricin inhibit EF2, blocking protein synthesis. Practice questions with answers. Previous year question, gate ecology and evolution. Q1, which elongation factor is responsible for delivering aminoacyl tRNA to the ribosome in prokaryotes? A. EFG. B. EF2C, EFTC, EIF2. Answer, B. EF2. Multiple choice questions MCQs Q2, what is the function of the Shine-Dalgarno sequence? A. Signals termination of translation D, recruits ribosomes to prokaryotic mRNA C, facilitates polyadenylation D, directs mRNA splicing. Answer, B. Recruits ribosomes to prokaryotic mRNA. Q3, which initiation factor is responsible for preventing premature binding of the large ribosomal subunit in prokaryotes? A. IF1. B. IF2C, IF3D, EIF4E. Answer, C. IF3. This detailed PhD entrance study note covers ribosome structure, initiation complex formation, initiation factors, elongation, and elongation factors, along with previous year and practice questions. Let me know if you need any further clarifications. You said. Termination, genetic code. Aminoacylation of tRNA, tRNA identity, aminoacyl tRNA synthetase, ChatGPT said. Protein synthesis, termination, genetic code, and tRNA aminoacylation 1. Termination of translation. 1.1. Signals for termination. Translation ends when the ribosome encounters a stop codon in the mRNA. There are 3 stop codons. UAA, ochre, UAG, amber, UGA, opal. These codons do not code for any amino acid and are recognized by release factors RFs instead of tRNAs. 1.2. Termination process recognition of the stop codon. Prokaryotes. Release factor 1 RF1 recognizes UAA and UAG. Release factor 2 RF2 recognizes UAA and UGA. Release factor 3 RF3 GTPase that facilitates RF1 RF2 activity. Eukaryotes. ERF1 recognizes all 3 stop codons. ERF3 GTPase that enhances ERF1 function. Hydrolysis of the peptidyl tRNA bond. The peptide chain is released from the tRNA in the P site. Dissociation of the ribosome. Ribosomal subunits, mRNA, and release factors disassemble for future translation rounds. Regulation. Nonsense-mediated decay NMD degrades mRNAs with premature stop codons to prevent production of faulty proteins. 2. The genetic code. 2.1. Features of the genetic code. Triplet code. Each amino acid is specified by a codon of 3 nucleotides. Degeneracy. Multiple codons can encode the same amino acid. Universal. The same codons specify the same amino acids across almost all organisms. Non-overlapping. Each nucleotide is part of only one codon. Unambiguous. Each codon codes for only one amino acid. Wobble hypothesis. The third base of a codon can tolerate mismatches, allowing flexibility in tRNA binding. Example, GU wobble pairing. 2.2. Start and stop codons. Start codon, AUG methionine in eukaryotes, N-formylmethionine in prokaryotes. Stop codons, UAA, UAG, UGA. 2.3. Exceptions to the universal code. Mitochondria have slight variations, e.g., UGA codes for tryptophan instead of a stop codon in mammalian mitochondria. Some protists use alternative codons. 3. Aminoacylation of tRNA, tRNA charging, 3.1. Overview. Aminoacylation or charging attaches an amino acid to its corresponding tRNA, forming aminoacyl tRNA. This process is catalyzed by aminoacyl tRNA synthetases, AARSs. 3.2. Steps of aminoacylation: activation of amino acid. The amino acid reacts with ATP, forming aminoacyl AMP. Enzyme, aminoacyl tRNA synthetase. Reaction. Amino acid plus. ATP right pointing arrow. Aminoacyl AMP plus. PPI. Amino acid plus ATP right pointing arrow, aminoacyl AMP plus PPI. Transfer to tRNA. The aminoacyl group is transferred to the 3'OH CCA tail of tRNA. Reaction. Aminoacyl AMP plus. tRNA right pointing arrow. Aminoacyl tRNA plus. AMP. Aminoacyl AMP plus tRNA right pointing arrow, aminoacyl tRNA plus AMP. 3.3. Function of aminoacyl tRNA: carries the correct amino acid to the ribosome. Forms peptide bonds with the growing polypeptide chain. 4. tRNA identity and recognition. 4.1. Structure of tRNA. tRNAs have a cloverleaf secondary structure and an L-shaped tertiary structure. Key regions include: anticodon loop, recognizes the mRNA codon. Acceptor stem. 3. CCA tail, binds the amino acid. D loop and T-C loop, provide structural stability and interact with ribosomal components. 4.2. Determinants of tRNA identity. Anticodon sequence, recognizes the correct codon in mRNA. Acceptor stem nucleotides, specific sequences help aminoacyl tRNA synthetases recognize the correct tRNA. Post-transcriptional modifications, example, inosine I in the anticodon enhances wobble pairing. 5. Aminoacyl tRNA synthetases, AARSs. 5.1. Types of aminoacyl tRNA synthetases. Class 1. Attaches the amino acid to the 2' of the ribose, later shifts to 3'. Example, glutamyl tRNA synthetase. Class 2. Directly attaches the amino acid to the 3' of ribose. Example, serine tRNA synthetase. 5.2. Fidelity of aminoacyl tRNA synthetase is highly specific for both amino acid and tRNA. Some enzymes have proofreading activity. If an incorrect amino acid is attached, the enzyme hydrolyzes the bond, preventing errors in translation. Example, isoacyl tRNA synthetase rejects mischarged valine. Practice questions with answers. Previous year question, Gate Ecology and Evolution. Q1. Which of the following recognizes stop codons during translation in prokaryotes? A. EFG. B. RF1 and RF2. C. ERF1. D. IF3. Answer, B. RF1 and RF2. Multiple choice questions, MCQs. Q2. Which enzyme is responsible for charging tRNA with an amino acid? A. RNA polymerase. B. Peptidyltransferase. C. Aminoacyl tRNA synthetase. D. EF2. Answer, C. Aminoacyl tRNA synthetase. Q3. What is the function of the genetic codes degeneracy? A. Allows multiple amino acids to be encoded by a single codon. B. Enables a single amino acid to be encoded by multiple codons. C. Prevents translation errors. D. Facilitates ribosome recycling. Answer, B. Enables a single amino acid to be encoded by multiple codons. This comprehensive PhD entrance study note covers translation termination, genetic code, tRNA aminoacylation, tRNA identity, and aminoacyl tRNA synthetases, along with previous year and practice questions. Let me know if you need further clarifications. You said, and translational. Proofreading, translational inhibitors, post-translational modification of proteins. ChatGPT said. Protein synthesis, translational proofreading, inhibitors, and post-translational modifications. 1. Translational proofreading. 1.1. Importance of translational accuracy. Translational proofreading ensures that the correct amino acids are incorporated into the growing polypeptide chain. Errors in translation can lead to non-functional or toxic proteins. 1.2. Mechanisms of translational proofreading. 1.2.1. Proofreading by aminoacyl tRNA synthetases. Editing site in synthetases. Some aminoacyl tRNA synthetases have an editing domain that hydrolyzes incorrectly charged amino acids. Example, isoacyl tRNA synthetase rejects mischarged valine. 1.2.2. Proofreading by ribosome. Kinetic proofreading. The ribosome delays peptide bond formation until correct codon-anticodon pairing is verified. Elongation factor 2. EF2. Prokaryotes. EF1A. Eukaryotes binds aminoacyl tRNA and only releases it if codon-anticodon pairing is correct. Incorrectly bound tRNAs are released before peptide bond formation. 1.2.3. Ribosomal decoding center. The 16 srRNA prokaryotes and 18 srRNA eukaryotes check for correct base pairing. Mismatch recognition triggers GTP hydrolysis delay, allowing incorrect tRNAs to dissociate. 2. Translational inhibitors. 2.1. Antibiotics targeting translation. Prokaryotic translation inhibitors used as antibiotics. Streptomycin binds the 30S subunit, causing misreading of mRNA. Tetracycline prevents tRNA binding to the A site. Chloramphenicol inhibits peptidyltransferase activity of the 50S subunit. Erythromycin binds the 23 srRNA of the 50S subunit, blocking elongation. Gyromycin mimics aminoacyl tRNA and causes premature chain termination. Eukaryotic translation inhibitors used in research or toxins. Cyclohexamide inhibits peptidyltransferase in the 60S ribosomal subunit. Diphtheria toxin inactivates EF2, blocking translocation. Ricin cleaves 28 srRNA, preventing translation. 3. Post-translational modification (PTM) of proteins. 3.1. Importance of PTMs. Post-translational modifications regulate protein stability, activity, localization, and interactions. 3.2. Types of post-translational modifications. 3.2.1. Covalent modifications. Phosphorylation. Addition of phosphate group co-cubed to ester, THR, or TR residues. Enzyme, kinases reversed by phosphatases. Function, regulates signaling pathways e.g., MAPK, insulin signaling. Acetylation. Addition of acetyl group coached to Li residues. Example, histone acetylation regulates gene expression. Methylation. Addition of methyl groups CH to Li or ARG residues. Function, controls epigenetic regulation e.g., histone methylation. Ubiquitination. Addition of ubiquitin to Li residues. Function, marks proteins for proteasomal degradation via the ubiquitin proteasome pathway. Sumolation. Addition of sumo, small ubiquitin-like modifier, to proteins. Function, alters protein localization and stability. 3.2.2. Structural modifications, disulfide bond formation between cysteine residues to stabilize protein structure. Example, insulin maturation requires disulfide bond formation. Proteolytic cleavage. Proteins are activated by removal of specific segments. Example, proinsulin right pointing arrow insulin after cleavage by proteases. Lipidation, membrane targeting. Myristoylation and palmitoylation, attach fatty acids to proteins for membrane anchoring. Example, ROS protein lipidation is essential for its function in cell signaling. Glycosylation, carbohydrate attachment. N-link glycosylation, adds sugar to asparagine ASN in the ER. O-link glycosylation, adds sugar to serine SR or threonine THR. Function, affects protein stability, folding, and immune recognition e.g., blood group antigens. Practice questions with answers. Previous year question, Gait ecology and evolution Q1, which of the following antibiotics inhibits peptidyltransferase activity in prokaryotes? A. Tetracycline. B. Streptomycin. C. Chloramphenicol. D. Cyclohexamide. Answer, C. Chloramphenicol. Multiple choice questions MCQs. Q2, which enzyme removes phosphate groups from phosphorylated proteins? A. Kinase. B. Phosphatase. C. Ubiquitin ligase. D. Peptidyltransferase. Answer, B. Phosphatase. Q3, ricin inhibits translation by targeting which ribosomal component? A. 30S subunit. B. 50S subunit. C. 28SRRNA. D. EFG. Answer, C. 28SRRNA. This detailed PhD entrance study note covers translational proofreading, inhibitors, and post-translational modifications with previous year and practice questions. Let me know if you need further elaboration. You said. Control of gene expression at transcription and translation level, regulating the expression of phages, viruses. ChatGPT said. Control of gene expression at transcription and translation levels 1. Introduction to gene expression regulation. Gene expression regulation ensures that genes are expressed at the right time, place, and amount in response to environmental and cellular signals. This regulation occurs at transcriptional and translational levels. 1.1. Levels of gene regulation. Transcriptional regulation controls mRNA production. Post-transcriptional regulation includes mRNA splicing, stability, and transport. Translational regulation affects ribosome binding and protein synthesis rate. Post-translational regulation modifies protein function through phosphorylation, ubiquitination, etc. 2. Transcriptional regulation of gene expression. 2.1. Regulation in prokaryotes. 2.1.1. Operon model, Jacob and Monod, 1961. Operons are clusters of functionally related genes regulated together. Types of operons. Inducible operon, e.g., lac operon, activated in the presence of an inducer. Repressible operon, e.g., TRP operon, turned off in the presence of a corepressor. Example, lac operon, inducible system. Regulates lactose metabolism in E. coli. Components. Latch, LACI, lac structural genes encoding beta-galactosidase, permes, transacylase. LACI, repressor gene, binds to the operator to inhibit transcription. Inducer, allolactose, binds to LACI, inactivating repression. Example, TRP operon, repressible system, regulates tryptophan biosynthesis in E. coli. High tryptophan, TRP acts as a corepressor, activating the TRP repressor to block transcription. Low tryptophan, repressor is inactive, allowing gene expression. 2.1.2. Sigma factor regulation. In prokaryotes, different sigma factors control transcription initiation. Example. Sigma to the power of 7, housekeeping genes. Sigma cubed squared, heat shock response genes. 2.2. Regulation in eukaryotes. 2.2.1. Chromatin modifications, epigenetic regulation, histone acetylation. Histone acetyltransferases, HATs, add acetyl groups, right pointing arrow loosens chromatin, right pointing arrow activates transcription. Histone deacetylases, HDACs, remove acetyl groups, right pointing arrow tightens chromatin, right pointing arrow represses transcription. DNA methylation. Cysteine methylation at CpG islands represses transcription. Example, silencing of tumor suppressor genes in cancer. 2.2.2. Transcription factors, TFs, and enhancers. Transcription factors, TFs, bind to promoter enhancer sequences to regulate RNA polymerase activity. Enhancers, DNA regions that bind activator proteins to increase transcription. Silencers, DNA regions that bind repressors to inhibit transcription. 2.2.3. Hormonal regulation of gene expression. Steroid hormones, e.g., estrogen, testosterone, bind intracellular receptors that act as TFs. Peptide hormones, e.g., insulin, activate cell signaling pathways to regulate TFs. 3. Translational regulation of gene expression. 3.1. Regulation in prokaryotes riboswitches. mRNA structures that bind small molecules to regulate translation. Example, thiamine riboswitch in bacteria. Small RNAs, sRNAs. Bind to mRNA and block ribosome binding or promote degradation. 3.2. Regulation in eukaryotes. 3.2.1. MicroRNAs, mRNAs, and RNA interference mRNAs bind to mRNA, leading to degradation or translation inhibition. Example, miR21 suppresses tumor suppressor genes in cancer. 3.2.2. Regulation via